Primer designing tools must be validated. That's my view. And we have actually developed recently a new online primer designing tool, thoroughly tested it with experiments, made interesting observations, modified the program based on the observations we made, and tested again with a lot of experiments, and then published it. I will talk about this in a minute, but let's take the case of primers and probes from junctions. Now, there can be various types of junctions in DNA and RNA molecules. Let's consider a molecule here, like for example, and let's mark 3 different regions which are distant, but they could be brought together by various processes inside the cell, like this. Maybe A, B, C could be exons that are brought together, and these introns could be removed eventually, but I'll just show them just for ease of understanding. They could be spliced out, as you know, they are removed, and only this mRNA would be there in case of exon-exon junctions in the mRNA. Let's take both original sequence as well as the spliced one, put them side by side conveniently so that we can understand. So you could use a normal primer as well as a junctional primer to distinguish this mRNA and the original sequence, which could be the primary transcript or hnRNA or the genomic DNA. Now, when the primer designed from exon-exon junctions binds to in the mRNA molecule, that would not be able to bind to the other DNA, which has introns, for example. Why? Because it has an intervening sequence on the template. This portion is complementary to the other part of the previous junction, exon. So this binding you wouldn't expect. But what if you have the primer anyway binding only to one exon at its 3' end? So I would get a product from here between this primer and this primer here in the unwanted molecule as well as here. So my ability to distinguish this from this wouldn't work, isn't it? This depends a lot on the difference in the melting temperature of the 3' end, which is this. So the melting temperature of only this region of the primer, let's call it TM1, melting temperature 1, and the TM of the complete primer. That difference is critical. How much difference is okay when we design primers is critical, and that can decide success and failure of the goal of the experiment. This is just an example that we have taken, and we have done certain experiments and published a tool where we can, the tool is available at this URL. The goal is to design primers of all types, and particularly the junction primers, which are a little tricky. And before publishing this, however, we have designed primers, many of them, and used them in RT-PCR and RT-qPCR experiments. So let's go through what happened in our process. So initially we thought 7-degree difference between the melting temperature of one part of it or the complete melting temperature, which is usually bigger, larger melting temperature. The difference should be 7 degrees, and that's a lot of difference, so it should work, meaning this primer shouldn't bind when the TM difference is so much on the original sequence. It should only bind to the junctional region. This was our expectation, but when we tested by experiment, we found that we get a band corresponding to the product found by the junction primer at the right place, but we also get an additional product because of unwanted binding. That means in the original DNA where there is a intervening sequences, hence the product length is longer. All right. Now what happens if we change it? We did change, and we kept the threshold of difference at 11 degrees. Anything less than that is not acceptable. When we did that, we got only the right product. We did not get any other unwanted product. That means even if your solution is contaminated with this, you would only amplify this region, or you can detect whether this is present or not very specifically. And fortunately, we got this type of an insight during experimental validations early, and we were able to change this rule in the software that we developed. We also challenged the entire process by contaminating deliberately. The genomic DNA was added along with the RNA molecule, and we tested, and it worked well. So this tool, of course, can also be used for regular primers and probes like cloning or whatever other purposes you have. We request you to use them, and we also have certain information. If you find interesting, you can visit these websites where we compiled all the primer designing software of different types and then compared them. Two types of tables are available. And if you do use the tool, please do tell it to others if you like it, and cite the software and the article. Now I do have some related videos. If you are interested, you can take a look at them. Now I should acknowledge the colleagues who have helped us to develop this software, and also funding agencies, and thank you very much.